everybody. So my name is Alexis Peyroles. I'm the CEO of Ose Immunotherapeutics. Ose Immunotherapeutics is a listed biotech in France. We focus in immunotherapy, so both immuno-oncology and autoimmune disease. We have a strong background in our cluster in Nantes, which has been working in transplantation for the past 25 years. So from that, we leverage these academic collaboration with lab centers and university hospitals in order to find new, innovative, and first-in-class targets in both autoimmune disease and immuno-oncology. The strength of the company is clearly from target identification. We do have our own abilities in humanized preclinical models, abilities to build monoclonal antibodies, bispecifics, and then to go up to the first clinical stages. In the way, finding the right partnerships in order to expand the company and give to all the products their best chance. We do have 3 partnerships today with big pharma companies like Janssen Biotech, Servier, and Boehringer Ingelheim. Our lead asset is in phase 3 in non-small cell lung cancer. It's a combination of neoepitope, which is being developed post-checkpoint failure, where you have a strong unmet need. We do expect to have 4 products into the clinic by the end of this year. So that's a very important year for us in order to make sure that we launch these products and we get the initial clinical results in 2019 and 2020, which will be great catalyst for the company. And as I said, we do have already 3 partnerships. We do represent a potential of 1.5 billion euros in terms of milestones, plus royalties around 10%, coming from 3 pharma companies. And the idea for us, we are 34 people in the company, and we do have around 20 people in research, all immunologists with a very strong background and scientific knowledge in order to develop new first-in-class projects, mainly in immuno-oncology, focusing on macrophages and myeloid cells, because we believe that's the next wave in immuno-oncology, and in autoimmune disease, obviously, working also in inflammation, for instance. So clearly, the idea for us in immuno-oncology is to work where we have resistance to checkpoint inhibitor, PD-1, PD-L1, for example, through our product Tedopi, which is working on neoepitopes, or working on macrophages like OSE172, which is targeting SIRP-alpha on the SIRP-alpha CD47 pathway, product that has been licensed to Boehringer Ingelheim. And in autoimmune disease, as autoimmune disease and immuno-oncology are always very well connected, as you have sometimes the same targets. So we are able to produce either agonist or antagonist in order to down-regulate T cells or up-regulate Tregs for the autoimmune disease part. And we believe that the 3 partnerships that we have with pharma companies, the various collaboration, international collaboration that we have with scientific labs, validate the quality of the science and the quality of the researchers that we have in our team. So the portfolio that we have in our company, so you have Tedopi, which is being developed in non-small cell lung cancer. It's a phase 3. It's a combination of neoepitopes coming from 5 tumor antigens, and it's being developed post-checkpoint failure. Today, we know that PD-1, PD-L1 checkpoint inhibitor are the standard of care in first-line and second-line non-small cell lung cancer, but clinicians need to have other options after checkpoints, and the kind of sequential combination that we are developing with Tedopi could be something of strong interest. The phase 3 is running in Europe, in the US, and in Israel, and we expect to have preliminary results by the end of 2019. As there is a strong rationale to combine Tedopi priming T cells with a PD-1 checkpoint inhibitor, GERCOR, which is a group of clinicians, they want to develop a phase 2 exploratory in advanced pancreatic cancer, combining Tedopi plus a PD-1 checkpoint inhibitor in order to see how we can enhance the activity of the PD-1 checkpoint inhibitor in this difficult indication. OSE172, so it's an antagonist of SIRP-alpha on the SIRP-alpha CD47 pathway, licensed to Boehringer Ingelheim. We are in charge of launching the first phase 1, which will be launched by the end of this year in Europe in solid tumors. And then Boehringer Ingelheim will take over all the development of the product. And we have a research collaboration with the Memorial Sloan Kettering Hospital in New York on one of our monoclonal, OSE703, targeting IL-7 receptor. And we work with Prasada Dusimuli, who is a specialist of CAR-T, and he wants to see preclinically for the moment whether adding our monoclonal or the sequences of our monoclonal to its CAR-T construct makes sense for the patients. It's a way for us to enter the CAR-T space with our own product without having to develop the technology by itself. In the autoimmune disease, we have two partnerships, one with Janssen Biotech, the other one with Servier, on two targets. The first one is CD28, which is a very interesting and strong target in the human body. We were able to produce a pure antagonist of CD28, and Janssen Biotech, after a phase 1 that we did in 2016, is now with a phase 2 status product working on further development. OSE127, which is under option license with Servier, is an antagonist of the IL-7 receptor, so the fuel of the T cells, and we are going to start the phase 1 by the end of this year, and we will develop it in ulcerative colitis, while Servier, after exercising the option, will develop it in Sjogren disease. So if we focus now on Tedopi, so Tedopi, it's a combination of 9 neoepitopes and a T helper. They come from 5 tumor antigens. They have been modified to have a very strong binding either to the MHC or to the TCR in order to enhance the T cell response. There is a clear rationale to combine them, this product with checkpoint inhibitor. And we are now in the phase 3 after checkpoint failure. So the product is given either in second line or in third line after failure of PD-1 or PD-L1. And as I said, we have also a planned phase 2 in pancreatic cancer with GERCOR. The idea is that neoepitopes are an answer, we believe, to the resistance to checkpoint inhibitor. We were able, by modifying these epitopes, to have a strong binding either to the MHC or to the TCR in order to obviously restore the immunosurveillance of cancer cells in HLA-A2 positive patients. That's the patients population where our product works. We have demonstrated that we induce T cell memory response, and we have obviously a strong patent family and an orphan status in the US. Non-small cell lung cancer is and remain a very big unmet need and a very big market. Checkpoint inhibitors have changed the landscape. Nevertheless, they still work for 20–25% of the population. So you need additional options after the failure of these treatments, and that's why we have a strong feedback from clinicians and a strong interest in our current phase 3 in order to give options, additional options to the patients, and Tedopi is one of these options. The resistance to PD-1, PD-L1 is being studied a lot lately, and one of the suspected mechanisms is the HLA downregulation. What does Tedopi is that it helps through interferon gamma upregulate HLA, and as such has an effect on the patients and on the disease. We have been interested by the phase 2 results that we have seen in these HLA-A2 positive patients. We had seen a strong correlation between the response to the various epitopes and the survival. And we also had been very interested to see that for the patients who were treated with Tedopi more than 2 years, we could see that 25% of these patients were still alive after 4 years, knowing that 67% of them were metastatic, that they had received at least 3 lines of treatment, and we even had 9% of brain metastasis patients in this study. So the current clinical trial is being developed right now in Europe, in US, and in Israel. We will have the first results on 100 patients by the end of 2019. What we want to see for this first step is whether we reach a certain percentage of patients still alive at 12 months. If we reach this level, which has been agreed with the agencies and the clinicians, we will enroll a total of 325 patients and compare this monotherapy of Tedopi versus docetaxel and pemetrexed, which is today what is used post-checkpoint failure. As I mentioned, we also had a strong interest from GERCOR, a group of collaborative group of clinicians, in order to combine Tedopi and a PD-1 checkpoint inhibitor in pancreatic cancer after what is today the standard of care, FOLFIRINOX. The toxicity of FOLFIRINOX does not allow to receive this product for too long, so we need to see other options, and we need to see whether with Tedopi and a checkpoint PD-1 inhibitor, we have a stronger maintenance, longest maintenance period in order to see the survival also at 12 months. So that's the same endpoint than in the phase 3 study. As I said, coming from transplantation, we have a strong knowledge of macrophages and myeloid cells, and we know that's the next wave in immuno-oncology. So our first product has been licensed to a Boehringer Ingelheim. It's a pure antagonist of SIRP-alpha on the SIRP-alpha CD47 pathway. Boehringer was for us a very good, potential very good partner. They have a strong portfolio in immuno-oncology. They have the global execution capabilities, so they're able to give to this product its best chance in order to develop it in Europe, in the US, and in Asia, in various indications, starting with solid tumors. And for us, obviously, the deal in terms of financial was very important because we received 15 million upfront. We are to receive additional 15 million when we enter phase 1, and the global deal is more than 1 billion euros. So it's a structuring deal for us, helping us to have a financial visibility until the end of 2019. How does it work? It's blocking SIRP-alpha on macrophages, and as such, transforming, I would say, bad macrophages into good macrophages. And we have demonstrated that by doing so, we have an effect on the whole tumor microenvironment. So we turn cold tumor into hot tumors, and as such, we see that we have better M1 macrophages, we have more T cells, less Tregs, and more NK cells. As you know, there are several companies working on this axis, SIRP-alpha CD47. We believe that our approach is differentiated in the fact that when you block CD47, you will block also SIRP-gamma. And as such, you might prevent a T cell expansion. Our product, by blocking only SIRP-alpha, does not block SIRP-gamma, and as such, allows a more stronger T cell expansion, making it a good rationale to combine with PD-1, PD-L1. As I said, we have also a research collaboration with the Memorial Sloan Kettering on Ose 703, which is a cytotoxic monoclonal antibody targeting IL-7 receptor. The idea is through targeting this IL-7 receptor in specific indication like mesothelioma or non-small cell lung cancer, for example, we could have a good synergy with the CAR-T being developed by Prasad Adusumilli. So for us, obviously, it's a way to investigate OSE 703 in solid tumors. The idea is to identify the efficacy profile and to enter the CAR-T space, which could help us develop other technologies in the future. Sorry. Sure. So in autoimmune disease, we've got two products. The first one is an antagonist of CD28, which is licensed to Janssen Biotech. CD28 is a very interesting target, very strong target. We were able, through our knowledge, to produce a pure antagonist of CD28. We have demonstrated in a phase 1 that the product is completely safe, and we think that, and we have preclinical models demonstrating the interest in various autoimmune disease. So we will have a phase 2 starting and managed by Janssen Biotech. And the other product that we have is IL-7 receptor antagonist, under option license with Servier, which will be developed in inflammatory bowel disease, namely ulcerative colitis, but also by Servier in Sjögren disease. All our partnerships allow to finance the company. We have been listed since 2015 without the need to get more additional capital from the market. These partnerships are strong and important for us, and we will continue in the future with our new products to get additional partnerships. That's the business model of the company. And we do have also either academic or public companies helping us in order to develop the company. So in order to finalize all this, we have a major immuno-oncology programs addressing unmet needs, Tedopi in phase 3 after checkpoint failure, in pancreatic cancer combining with checkpoint PD-1. We have one product entering phase 1, OSE172, targeting macrophages, and we will have also OSE127 in autoimmune disease, which will enter phase 1 by the end of this year, while FR104 is managed by Janssen Biotech for a phase 2 status, and we will have obviously our new research project that will be disclosed in 2019. Thank you very much.
Thank you, Alexis. Any questions?
Thank you.